Welcome back to this next video, and in this video we are going to talk about the population genetics. Now, what is population genetics? The population genetics, it can be defined in multiple ways, and one of the way in which you can define this is that it is a field of biology that studies the genetic composition of a biological population. Usually when we talk about the genetics, we usually talk about individuals, but in population genetics you are actually studying the genetic composition of biological populations and the changes in genetic composition that result from the operation of various factors, including the natural selection. So the difference between the population genetics and the general genetics, you can say that generally we focus on individuals, but in population we are going to focus on biological populations. Another way you can define a genetic population is that it is a subfield of genetics. As I've told you, that in genetics we are usually focusing on individual, but in population genetics, which is a subfield of genetics, that deals with genetic differences within and between populations. For example, if you are talking about the population of a city, that means you are actually dealing with genetic references within that particular city. But if you are comparing one country, the population of one country and the population of other country based on their genetic differences, that will also be covered in the population genetics. So in population genetics, they usually deal with genetic differences within and between population, and population genetics is usually considered as part of an evolutionary biology. Now, some of the important terms that you need to understand about the population genetics are that what is a population. Now, all of the inhabitants of an interbreeding species, now this is important, that all of the inhabitants of an interbreeding species means that they can interbreed with each other within a particular place and a particular time. That is known as a population. If you specify this, you can actually define this sexual population, which is a set of the organism in which any pair of members can breed together. Now, this implies that all members belong to the same species and they live near each other. For example, if this is a population of the deer, for example, they belong to the same species and they are actually interbreeding with each other. So you can consider this as a population, and you can also consider this as a sexual population. Another important term, that is the gene pool. Now, this term is typically used in reference to a population made up of individuals of the same species and include all genes and combination of genes. So when you are focusing on all genes and combination of the genes, that is sum of all the alleles in a population, we usually use this term gene pool. So gene pool actually refers to all the genes and combination of genes in a population. Like, for example, if this is a population and all of the genes and its combination, they are going to make the gene pool for this particular population. Another important term, that is the allele frequency. So what is allele frequency? The allele frequency is the fraction of the genes in the pool that is composed of that particular allele. Just give you a simple example, that if this is a population, and if you are focusing on the gene for the color of the flower, so one of the allele of the color gene that codes for the that is purple one, and another allele that is a white one. So if you are determining that in this particular population, what is the frequency of the purple allele and what is the frequency of the white allele, that is usually known as the allele frequency. So in this particular population, you are going to find out that how many alleles they are purple and how many alleles they are white. So you are actually focusing on alleles over here. One of the important equation about the population genetics is known as the HardyWeinberg Principle. It is also known as the HardyWeinberg equilibrium, or the HardyWeinberg model or theorem or law. all of them, they are actually referring to one and the same thing. So you can simply call it is the Hardy-Weinberg principle. Now this principle was given by Hardy, who was an English mathematician, and Weinberg, who was actually a gynecologist. So they gave a principle and they defined this as that the allele and genotype frequencies in a population will remain constant from one generation to another generation in the absence of the forces that changes it. What they mean to say is that if these forces they are absent, we will discuss the forces at the end of this lecture. So that if these forces they are absent, the allele and the genotype frequencies they will be constant in a particular population from one generation to another generation. Two terms they are important over here. One that is the allele and the other one that is the genotype. So you should differentiate between the allele and the genotype. For example, if you talk about this one, this capital W, it will be allele, the small w, it will be allele. So when they are written separately, that means you are talking about the allele. But when they are written together, like capital W, the capital W or the small w, the small w or the small w, this is actually known as the genotype. So what they say is that the allele and the genotype frequencies they will be constant in a population from generation to generation, provided that the forces that can change it they are absent. Let me give you a very simple case to understand the Hardy-Weinberg principle. For example, we take a gene, a single locus, which is made up of two alleles. There is one gene and there are two alleles. We denote one of the alleles with the capital A and the other allele with the small a. So usually when we use the capital letter, we usually represent a dominant trait by this, and if we use a small letter, we actually use it for the recessive allele. So you have got a single gene and you have got two alleles. So if you talk about the frequencies of these allele, you can actually represent that by this. F is for the frequency, this is the frequency of the capital A. So let us represent the frequency of the capital A by small p. Let us represent the frequency of the small a with the small q. Now these are the alleles. You are talking about the allele frequencies. When you talk about the genotypes, what you can expect is that the capital A, it can combine with another capital A, which will be making the A homozygote. So if you are representing the capital A with a small p, so you will be representing the capital A and the capital A with the p square, and it is going to represent the capital A homozygote. If you are talking about the small a, if you are using that to represent the small q, so if you are actually using the q square, that will be a and the a. Again, this q square or the aa, that is going to represent the aa homozygote. If you talk about the heterozygote, in which there is one capital A and there is one small a, you can actually represent that by the p and the q. We will come to this too in a while. So if you talk about the alleles, the capital A allele can be represented by the small p, its frequency, and the frequency of the small a can be represented by the small q. And if you are going to convert that into the genotype frequencies, what you can actually do is that if you are using the small p for one capital A, so if you are using a double capital A, you will be using the p square. For double small a, you will be using the q square, and for one capital A and one small a, you will be using the 2pq. Now the genotype proportion, this p square, this 2pq, and this q square, this is actually called is the Hardy-Weinberg proportion. Now if you note that sum of all genotype frequencies of this case is the binomial expansion of the square of the sum of the p and the q. Like for example, if you are adding the p plus q and if you are taking a square root, if you are taking a square of this, that is actually going to show you the binomial expansion of all of the genotype frequencies. And if you're talking about population, so all of the genotype frequencies, they are usually equal to 1 or they are equal to 100%. So this particular equation, this p plus q whole square, which is equal to p square plus 2pq plus q square in a population, that will be equal to 1 or that will be equal to 100. So if you look at this one, this is actually covering all of the possible genotypes in a population. This p square is going to cover the homozygotes, the dominant homozygote. This 2pq is actually going to cover the heterozygote, and this q square is actually going to cover the homozygote of the recessive trait. So all of these traits, they will be equal to 1 or they will be equal to 100 in a population. Now let me clarify all of this equation by using an example. Now, there is a particular chemical which is known as the phenyl thiocarbamide, which is also known as phenyl thiourea. That is an organosulfur thiourea containing a phenyl ring. Now this is the structure of the PTC, or the phenyl thiocarbamide. Now this PTC, it have got an unusual property, is that it either tastes very bitter or it is virtually tasteless, depending upon the genetic makeup of the taster. So if you are a taster for this particular PTC, you can actually you will look like this, because it is very bitter in nature. But if you are a tasteless for this particular PTC, you are not going to feel anything. And the this PTC, when it is usually very bitter or it's tasteless, it totally depends upon the genetic makeup of the individual. Now, the ability to taste PTC is a dominant genetic trait. That means that if you have got a dominant genetic trait for the PTC, you would feel this very bitter. Now, let us take you can say a population. As I've told you that this taster phenomena, that is a dominant one, and the non-taster phenomena, that is a recessive one. We represent the taster allele by the capital T, and we represent the recessive allele by the small t. If we have a population, for example, in which there are 50% tasters and there are 50% non-tasters, as I've told you that the taster phenomena, that is a dominant one, that means that the taster phenomena can represent itself in the homozygous condition, and it can also represent itself in the heterozygous condition. So this 50%, that is these individuals actually can have this particular genotype, or they can actually have this particular genotype. If you talk about the non-tasters, they can only have one genotype, that is the small t and the small t, because we all know that the recessive allele, when the condition that is of the complete dominance, the recessive allele only express itself in the homozygous condition. So the tasters can be in the homozygous or heterozygous conditions. The non-tasters are only in the recessive condition. Now, the possible marriages in this particular population, you can actually cross homozygous dominant, a homozygous taster to a homozygous taster. You can cross a homozygous taster to a homozygous non-taster, and you can have many different options for possible marriages in this particular population. If you can see over here that if we say that 50% they are tasters and 50% they are non-tasters, what you can do is that if you talk about the sperms, 50% of the sperm, they are going to carry the capital T allele, and 50% of the sperm, they are going to carry the small t allele. Same will be true for the eggs, that 50% of the eggs, they will be carrying the capital T allele, and 50% of the eggs, they are going to carry the small t allele, because they are 50%. Now, if you cross them, so if you are going to cross this particular sperm with this particular egg, what you can actually do is that what you will be getting is you will be having a capital T from this particular parent and a capital T from this particular parent. So these individuals, they are going to be homozygous tasters. What percentage? If you cross 0.5 into 0.5, that is going to give you 0.25%. If you cross this particular sperm with this particular egg, what you can actually get is capital T from this sperm, the small t from this egg, and if you cross 0.5 to 0.5, you'll be having 0.25. Again, these individuals, they will be tasters, but they will be heterozygous tasters. Similarly, if you cross this with this one, the capital T from here, the small t from here, and if you cross 0.5 into 0.5, again that will be 25 or 0.25 or 25% of the whole population, and again they will be heterozygous tasters. If you cross this with this one, the small t from here, the small t from the sperm, and when they combine this particular portion of the population, they will be non-tasters. So if you talk about the complete population, 25% they are homozygous tasters. Like this is 0.25, this is 0.25, this is 0.25. 0.25 means all of them, they are equal, they are representing 25% each. So 25% are the homozygous tasters, as you can see over here. 50% they will be heterozygous tasters. This is 25%, this is 25%. So 50% they will be heterozygous. And if you talk about this one, 25% they will be homozygous non-tasters. So totally in this particular population, 75% they will be tasters, irrespective whether they are homozygous or heterozygous. But in the whole population, there is 50% and 25%. 75% will be tasters, and 25% they will be non-tasters. Now, these particular, they are actually giving you the genotype frequencies, that 25% will be having this particular genotype, 50% will be having this particular genotype, and 25% they will be having this particular genotype. Now, if you talk about the allele frequency, that what will be the allele frequency, by that I mean that what will be the frequency of the capital T allele and what will be the frequency of the small t allele. If you look at this one, the capital T allele, it can come, if you talk about this particular population, they are 25% of the whole population, and they are only reproducing one allele. They are actually having only one allele, that is the capital T. So 25% of the capital T allele, that will come from this particular population. If you talk about this one, which are the 50% heterozygous tasters, they are having the capital T allele as well as the small t allele. So if the capital T and the small t, and this capital T and the small t, if they are representing 50% of the population, that means that if you are making sperms, 25% will be having the capital T allele and 25% will be having the small t allele. So here it is 50%, but when you focus on a single allele, that means that 25% capital T allele, that will come from this particular heterozygous population. So this capital T and this small t, they are actually making 50% of the whole population. But when you talk about the specific allele, only 50% from this particular population, they will be having the capital T allele. So this is 25% from the capital T, and this capital T, it can come from the capital T and the small t. So this 25% and this 25%, that is actually going to give you 50% of the capital T allele frequency. Similarly, when you talk about the small t allele, 25% that will come from this particular population, and similarly 25% that will come from this particular population. So that is again going to make 50% of the small t allele. So if you look at here, the genotypic frequency and the allele frequency, they are actually two different things. Here you are actually having 75% tasters, but when you talk about the allele frequency, only 50% of the allele that are for tasters, and 50% of the allele they are for the non-taster allele. So as the Hardy and Weinberg say that if the conditions, if the forces they are absent, the genotypic and the allele frequency they will be constant from one population to another population. So if you look at over here, 50% again will be capital T allele, 50% will be small t allele. Again you will be getting this particular conformation. So if you cross them again, you will be getting 75% tasters and 25% non-tasters. Now how you are actually determining the allele frequency using the particular equation of p square plus 2pq plus q square. Now let us take an example of a population in which 70% are tasters and 30% are non-tasters. If you look at these tasters, you actually cannot determine the allele frequency from here directly, because the tasters they can be in the capital T or capital T genotype, or they can be in the capital T or the small t genotype. But when you talk about the non-tasters, they can only be present in the small t and the small t genotype. Now we will use this information, this 30% non-taster information, to determine the allele frequency of the capital T and the small t. Now as we know that the p square plus 2pq plus q square, that is equal to 1, that means that all of the genotypic frequencies, when you sum them, that is going to be equal to 1. Now we are given that the non-tasters they are 30%. We know that the non-tasters, we will represent them by the q square. So this q square is equal to small t and the small t, and this genotype is actually representing the non-taster phenotype. We are given that the non-tasters they are actually 30% of the whole population. So if you are talking about the 1, so 30% will be equal to 0.3. Now as we are interested in the allele frequency and we are yet not interested in the genotype frequency, so what we can do is that if we take the square root at both sides, the square root of this side and the square root of this side, so that will give us the if we take the square root of the q square, that is going to give us the q value. If we take the square root of the small t and the small t, that is going to give us the small t. And if you take the square root of 0.3, that is going to give you a value of 0.547. I have already made the calculation, so you should practice this, how this actually works. Now as we know that the this q, that is one allele, but this p, that is another allele, so all of the allele that will be equal to 1. Now if we are interested in determining the value of the p allele, so what we will do is that we can actually do this p, so that this q, when it shifts to the other side of the equation, it will become p is equal to 1 minus q. This is p again 1. We already have calculated the value of the q, which is 0.547, so we will put it over here. So again this is going to give us the allele frequency of the capital T allele, or the p, which is actually the 0.46%. Now if you want to confirm this, whether our values they are correct or they are not correct, so we can actually do this by again determining the genotypic frequencies. So we have determined the value of the q, we have determined the value of the p. Now we want to determine the value of the 2pq. So what you can do is 2pq is equal to 2, the value of the q is 0.547, the value of the p is 0.46, and when you calculate this, that is going to give you the value of 0.5. Now if you look at the equation, this particular equation, the p square plus 2pq plus q square, that is equal to 1. So the q square, we have already been given that the value of the q square, that is 0.3. So we will put this at the 0.3. We have determined the value of the 2pq, which is actually the heterozygous condition, and we have know that the value is 0.5, and the value of the p square, if you add 0.5 and 0.3, that will be 0.8, and if you minus the 0.8 from 1, the remaining value that will be 0.2. Now if you look at over here, this one, this 2pq, that is the taster condition, but in the heterozygous form. The p square, the capital T, that is the taster condition, but in the homozygous form. So this is actually making 70% of the population, as 0.5 and 0.2, that is making 0.7%. So out of 1.7 actually makes 70%. So we are having the 30% non-taster, 70% tasters, which is the situation that is given to us. And if you determine the allele frequency from these particular values, you can actually see is that the small t, that is 0.3 over here, as you can see over here. The value of the this small t is also present over here. So this heterozygous condition, it is total of 50%. That means that out of this 50%, 50% will be the small t, and 50% will be the small t, and 50% will be the capital T. But at this is 0.5, so if you divide this by 2, that means 0.25 that will be for the small t, and 0.25 that will be for the capital T. So this small t 0.3, it comes from here, and from this 0.25 comes over here. So the allele frequency of the small t, that is 0.55, and the allele frequency of the capital T, that will be 0.5 over here. Now if you can see over here, the allele frequency of the capital T, that is very low, that is low as compared to the allele frequency of the small t, but as the capital T can express itself both in the homozygous and the heterozygous condition, so in the population the tasters they are more as compared to the non-tasters. Let me give you another example, and this example is very important if you are taking the ETA test for your medical college. Now if you are given if there is a certain population of rabbits in the Midwestern United States, and there are two alleles for the coat color of the rabbit. One of the allele that codes for brown, and one of the allele that codes for the white coat color. So you have got a single gene for the coat color, but that single gene have got two alleles. One allele is for the brown color, and one allele that is for the white color. This is one condition that is given to you, that there is one gene but there are two alleles, one brown allele and one white allele. Another information that is given to you in this question is that the brown allele that is dominant. This means that the brown is going to represent itself in the homozygous as well as in the heterozygous condition, and the white that is recessive. That means it can only represent itself in the homozygous condition. It is not able to express itself in the heterozygous condition. Now what you have to do is, another information that is given to you is that the frequency of the white colored rabbit, that is 9% of the total population. So three things they are given to you, that there is one gene but there are two alleles, the brown and the white allele. The brown allele is dominant and the white allele is recessive. And another important information is that in this particular population, the frequency of the white colored rabbit, that is 9%. Now the first question they ask you is that what is the frequency of the allele for the brown coat color in the rabbits. Now we have to determine the allele frequency of the brown allele, that how much brown allele is present in this particular population. Now what is given to you is that the white color rabbits, they are actually making 9% of the population. So you can actually see is that the white color rabbits, they are making 9% of the population, and as I've told you, as the white is recessive, so we are going to represent the white allele by the small b. The brown color rabbits, if these two are making the population, so if 9% they are white, so 91% of the rabbits, they will be brown. So as the brown is dominant, so we represent the allele by the capital T, by the capital B, not capital T. Now as we know that this is the Hardy-Weinberg equation, that p square plus 2pq plus q square, that will be equal to 1. Now here we are interested in determining the allele frequency, so we are going to determine the allele frequency, and we are going to remove this genotype marks for a while. So this q square is actually representing the recessive one, the recessive population over here, and as the recessive population is represented by the small b and the small b for the white coat color of the rabbit. So this q square will be equal to small b and the small b, and we have already been given the value that it is 9%. But as we are dealing it with 1 now, so if you convert the 9% out of 1, that is going to give you the value of 0.09. So this 0.09 or this 9%, that is one in the same thing, but this 9% is in 100 and this 0.09 is in 1, but they mean one in the same thing. As I've told you in the previous slides, if you are going to take the square root at both sides, what you are going to get is that instead of the q square, you are going to have the q. Instead of the small b and the small b, you will be having a single small b, and if you take the square root of 0.09, that is going to give you the value of 0.3. Now the p plus q, again, that will be equal to 1. If you are interested in determining the value of the small p, what you can do is that you can shift the equation, p is equal to 1 minus q, because if this q is shifted over here, so that will become 1 minus q. So p will be equal to 1. The value of the key we have already determined that, so this will be a 0.3. So the value of the p, that will be equal to 0.7. Now if you are determining the value of the 2pq, what you can actually do is that 2, this is the value of the p, the 0.3 is the value of the q. So if you multiply this, you are actually going to get the value of 0.42. Now if you talk about the genotypic frequencies now, that the q square, which is represented by the small b and the small b, that is actually making 9% of the population, or 0.09 out of 1. So this is 0.09 or 9% of the population. Now the value of the 2pq, so this all of these individuals that will be having, all of these rabbits, they will be having a white coat color. If you talk about the 2pq, that will be represented by the capital B and the small b. This is the heterozygous condition, is the brown that is dominant over the white one. So these rabbit, they will be having the brown coat. So the value of the 2pq, that is 42%. If you talk about the p square, as I've told you, this 42 plus 9, that is going to give you 51. So the remaining is 49%. So the remaining rabbits, they will be brown, but that will be homozygous brown. So the q square, that will be 9%, the 2pq, that will be 42%, and the p square, that will be 9%. Now if you talk about the allele frequency, that what will be the allele frequency of the brown one, we are actually interested in the allele frequency of the brown one. We have already seen it over here that the value is 0.7. But if you want to confirm this, these genotypic values, they are actually giving you the confirmation that the value you have calculated, that is okay. So from here, if you determine the allele frequency, as you can see that this small b 0.09, that will come from here. These rabbits, they will be producing only a single kind of the allele, that will be the small b allele, and they are actually representing 0.09 or 9% of the whole population. If you look at these individuals, they can produce the capital B allele, they can produce the small b allele, as this is 42%. So if you divide it by 2, that is going to give you the value of 21 or 0.21. So if you add the 0.09 and 0.21, that is going to give you the value of 0.3 that we have already determined for the q. The value of the b over here, if you can see over here, that this particular rabbits, they are making 49% of the population, and they are only making a single kind of the allele, the capital B and the capital B. So the 49%, the capital B, they will come from here, and these rabbits, they can produce the capital B and the small b allele. So as we have measured it for the small b, that 0.21, if you divide this 42 by 2, so 21, that will be for the small b, and the remaining 21, that will be for the capital B. So this is going to be a 0.7. So your answer, that the what is the frequency of the allele for the brown coat color in this particular population, your answer will be 0.7 or 70%, you can say. Now another question they can ask you is that in this population of rabbit, what percentage is hybrid brown? Hybrid brown mean the heterozygous condition. So as you have already made the calculations, you have already determined the genotype. So if you can see over here, the hybrid brown, they are 42%. So your answer, that will be that the 42% of the rabbits in this particular population, that will be a hybrid brown. Another question is, if they ask you that if the climate were to change, so that now snow covered the ground much of the time, what change in the population of rabbit would you expect? What this means is that the brown color is initially an advantage in one condition, and the white color is an advantage in another condition. So if there is snow, covered the ground, if the snow is covering the ground much of the time, that means that if the rabbits, they are producing the white coat color, they can actually prevent themselves from the predators. So if the climate change, so that now the snow covered the ground much of the time, what change in the population of rabbit would you expect? And these are your options. That the frequency of the white allele would increase. I have already highlighted this. This should be your answer, because this is going to give advantage to the rabbits, so that they can be prevented from the predators. The frequency of the white allele would decrease. That is not a correct answer, because if you are decreasing the white allele, that means that will be that the rabbits, if they are producing the brown coat color, they will be exposed to the predators like the wolves, and they will not survive then. The frequency of the brown allele would increase. Again, this is not a correct answer. The population of the wolves or the population would decrease and then increase. The population of the wolves would increase and then decrease. It has nothing to do with the color of the coat color of the rabbit. Now, as I've told you, that Hardy and Weinberg say that the allele and genotype frequency will be constant from one generation to another generation in a particular population if certain forces, they are absent. These are some of the forces which must be absent if the Hardy-Weinberg law is to come into action. Now, one of the forces is that the mating choice and the selection. What Hardy and Weinberg say, that the crossing between the species, the crossing between the members of the population, that should be selected. That the members of the population, they should not interbreed with each other without any selection. So if you are making the breeding a selective process, then you can actually maintain the allele and the genotype frequency. But as you cannot control the mating choice or the mating selection in a population, this particular force very badly affect the law of the Hardy and the Weinberg. Another force that should be absent, that is that there is no mutation in the population. But the Hardy and Weinberg say that our law will be applicable if there is no mutation in the population. Another thing, they say that there should be no gene flow, no migration from the population and into the population. The fourth point is that there should be no disease or natural disaster deleting a particular allele community, and all of the members, they should produce the fertile gamete. The Hardy-Weinberg say that if all of these forces, they are absent, then the allele and the genotype frequency, that will remain constant from one generation to another generation in a population. But if these forces, they are present, the allele and the genotype frequency, they will not be constant from one generation to another generation. We will continue the discussion in the next video.